Hey guys, welcome back. This time around we're going to continue our discussion of biochemistry, particularly looking at the organic molecules. Thus far we've looked at carbohydrates and lipids. This time around we're going to look at proteins. Proteins are going to be built from monomers known as amino acids. Proteins are going to represent anywhere from 10 to 30% of the cell mass. Proteins are going to be found in structural components of our body as well as key players in all the physiological activities that occur in our body. Great places in which you would find proteins would be in muscles, hemoglobin, enzymes, hormones, and much, much more. Proteins, as well as the amino acids, are going to be composed of some key elements. Those elements are carbon, hydrogen, oxygen, nitrogen, sulfur, and phosphorus. Let's look at those amino acids. Amino acids are going to be the basic monomers of all proteins. There's going to be 20 different types of amino acids found in our body. Now, these 20 different amino acids have some things in common. First and foremost, they have a centralized carbon, sharing 4 valence electrons. Attached to that carbon are going to be 3 side groups that every amino acid has. One of those side groups is going to be hydrogen. Another one of those side groups is something we call the amino group. Now, that amino group has a nitrogen in it as well as a hydrogen. It's actually going to be basic as far as pH is concerned. And the other side group is going to be a carboxylic acid, COO negative, which is going to give us an acidic side to it. Now, each of the 20 amino acids share these 3 side groups. What makes each amino acid unique is going to be what's found on the 4th side group. Now, we call that 4th side group either the R group or the functional group. Now, what can be found on that functional or R group could be something as simple as hydrogen. If it is, then we have an amino acid known as glycine. It could also be a side group that could make it polar. It could be polar and be a basic amino acid such as lysine or an acidic amino acid such as glutamic acid. There could also be nonpolar characteristics associated with that R group, as well as that R group having a side group associated with sulfur. Now, frequently we'll find these amino acids floating freely in our body. Now, as a group, these amino acids can be modified into something we know as amines. Now, amines frequently are going to be seen as key neurotransmitters in our body. But these amino acids also are chemically bonded together to build larger structures. If I take 2 amino acids and chemically bond them together, I create something known as a dipeptide. Now, the bond that's there is a covalent bond, but we give it a particular name. We call it a peptide bond. Now, if we take additional amino acids and chemically bond them together, we make something known as a polypeptide or an oligopeptide. By definition, in this class, a polypeptide is going to represent 4 to 49 amino acids chemically bonded together. Great examples of polypeptides would be the growth hormone releasing hormone, calcitonin, enkephalin, and glucagon. Now, if I take 50 or more amino acids and chemically bond them together, I make something we call a protein. Good examples of proteins include hemoglobin, insulin, growth hormone, enzymes such as lactase. And when we're looking at these proteins, we're going to find that proteins will have a variety of structures to them. The 4 key structures that we need to be able to identify include the primary structure. Now, the primary structure is just the sequence of amino acids. Realize that every protein is going to be unique in one. It has 50 or more amino acids chemically bonded, but it's also going to be unique in the order. Think of it, the only function of DNA, truthfully, is to decode for us the order of amino acids it takes to build each and every protein in our body. So the primary structure will tell us that proper orientation or order of amino acids, of the 20 different amino acids. Then we start seeing some folding interactions occurring between the amino acids, adjacent to one another. The initial folding we call secondary. Now that secondary can be one of 2 types. It can be an alpha helix. It looks sort of like a corkscrew. In this case, what we're seeing is an interaction between the amine of one amino acid and the carboxyl group of another amino acid. Typically, these interactions are at every 4th amino acid. The other type is known as a pleated sheet. A pleated sheet sort of looks like bed sheets, in which we see polypeptide chains lining up with one another. And they're either going in the same direction, in that case we'd call it a parallel or an alpha pleated sheet, or they're going in opposite directions, we'd know it as a beta pleated sheet or non-parallel. The third structure that we can see is taking these secondarily folded amino acids and having them fold upon themselves, creating something that we would think of as a tertiary. Now, in the case of a tertiary, it would look much like us taking, for instance, a piece of paper and turning it. Now, this twisting here, we would think of as secondary, and then if we take it and fold it further, we know that as tertiary. Tertiary conformation is going to be folding in the secondary, in which we see interactions that are hydrogen bonding interactions between amino acids, or disulfide, or sometimes even the nonpolar amino acid will interact. Now, if we see more than one tertiarily folded protein interacting with one another in space, then we know that as quaternary. Now, quaternary, the best example I can think of is hemoglobin. With hemoglobin, we've got 4 different tertiarily folded molecules interacting with one another in space to create the hemoglobin molecule. So hemoglobin molecule has 4 subunits, each subunit individually active, but as a cluster working together. They do that to save energy.